Right, good afternoon, everybody. It is a pleasure to see all of you here. Thank you for making time to come to our research break. My name is Susan Brown. I'm the Director of Research Coordination and Research to Practice Integration at the Schlegel University of Waterloo Research Institute for Aging. And I'm delighted to welcome Dr. Andrew Costa this afternoon. Andrew is the Schlegel Research Chair in Clinical Epidemiology and Aging at McMaster University and is co-leading the largest study in the world about vaccinations for COVID-19 in long-term care homes. So we're excited to have him here. Just a couple of housekeeping items before we begin. If you're having any technical issues, don't worry, you won't miss out on the content of the webinar. We are recording this and we'll send out a recording to everybody who's on the call by sort of early next week or so. We're going to have about 20 minutes of a presentation from Andrew and then 10 minutes for questions. On the right-hand side of your screen, you should see a little speech bubble with a question mark in it, and you can hit that and then type in your questions. Jasmine will be working in the background to help compile those questions, and if we don't get around to answering all of them at the end, then we'll be getting all of those responses together and publishing sort of a summary article on our website in the next little while. So please welcome me, or don't welcome me, but help me in welcoming Andrew this afternoon. And Andrew, I will pass it over to you.
Okay, thank you, Susan. And I hope that you can confirm that you can see the slide.
Looks to me like it's still loading.
Okay, I'll try something here. Okay, and then we'll try it this way.
That's good. I can see it now with your notes.
Okay, and can you see it in presentation mode?
No, I don't see anything at the moment.
Okay, folks listening, we're having a live glitch, but fear not. It looks like when I put it in presentation mode, somehow it doesn't. But we will probably might have to do it this way. I'll try again, and then you let me know what you see.
Okay.
Still blank, it looks like.
Still blank, yeah.
That's curious. We tested it, but no problem. You can see, I think, what folks need to see, so we'll begin. Thanks for inviting me, Susan, and to the RIA also for the invitation. Very much appreciated. Today I want to basically present in a short period of time essentially some of the information that was informative and responsible for the approval of the third dose, or the recommendation of the third dose for long-term care residents in Ontario, which has since been expanded to individuals in retirement homes as well as individuals in other congregate settings like assisted living, etc. And this is some results that continue to come in fact, from a study that I am co-leading with Dr. Don Bowdish. I'm an epidemiologist. Don Bowdish is an immunologist, and so together we came together to do this study together with our partners, and a very, very large team and very large cast of co-investigators who are listed on the slide, as well as partners. And this was funded by the COVID-19 Immunity Task Force for the country. Susan, you said that it was the largest in the world. I don't know that's the largest in the world. It's certainly a very large study in Canada, and the result of some of which I'll show you should be published shortly in JAMDA. So the vaccine in Ontario, that's really what we're looking at. When we started this study, we weren't looking at the vaccine in Ontario because the vaccine wasn't yet available, not announced. It was approved very quickly and obviously distributed across long-term care homes and indeed retirement homes in in the early part of the new year. And basically, it's been a game changer. I think it's pretty obvious, those working in long-term care, it's been a massive game changer, where it has reduced the rates of COVID-19 infections as well as deaths in long-term care substantially. And so this shows in panel A the relative reduction from 1, essentially, for residents, where the solid line is deaths and where the dotted line is infections over the first 8 weeks. An absolute massive relative reduction when at the same time infection rates in the community were essentially going up. And this is compared, in fact, to those who were not able to be vaccinated yet because they weren't part of phase 1, older adults who were matched in the community. And that's also true for staff, where we see a huge reduction in infections. There were no deaths to report, and so you see none here. And that's the first 8 weeks. Since then, Public Health Ontario and others have been looking at the rates of outbreaks. And as you see in the figure in front of you, the number of outbreaks sort of spike and they ebb and flow generally with the community infection rates around long-term care homes. And the red line there shows the start of the vaccination program, where you see a drop in outbreaks. Outbreaks being essentially any staff or resident infection, one or more of either. And what we observed in the same time was that even for those outbreaks, the size and scale and lethality of those outbreaks were changing rapidly. And this goes up to June, and essentially we've seen a relatively quiet infection picture in long-term care since then. Certainly much quieter than we could have otherwise expected, given outbreaks that existed amongst other in the community, other groups, etc. So it's been a real game changer. And so the mRNA vaccines have really worked, which these are the vaccines that have been offered to long-term care residents and most staff. And it's pretty clear that the vaccine-mediated immune response has really changed things. The bad news is that, and I'll sort of bury the lead now, is that the immune responses for long-term care residents, in any case, don't seem to last very long, or at least there is concerns around how long they do last, to the point where obviously we've have a third dose that's been recommended across the country. What do we mean by these doses? And so this is sort of the classic picture of an antibody-mediated immune response, where in the first dose, essentially what you're doing is priming the immune system to react, to recognize the virus by showing it either a dead copy or a feature, which is what the mRNA vaccines essentially do, of the virus, so that it knows how to attack it in the future. And then a second dose sometimes completes the response, where you have a full immune response to that second dose after the primer. And sometimes a third dose or more, but in vaccines, third dose is not rare, is required at some time, essentially, to provide a booster, to remind your immune system how to fight the virus. This is true for essentially most, if not all viruses, all vaccines, excuse me. When we think about immune responses, really our best bet is to compare them to healthier populations or other populations to see where folks are at, because the amplitude of these jumps are not really absolutes. And really we understand partial versus complete immune responses according to how well some populations respond compared to others. And so for long-term care residents, we like to compare them to other younger, healthier groups on the same basis using the same assays. And that gives us a sense as to where things are at. And essentially the concern is that sometimes that first prime might not be totally adequate. And sometimes the second dose for some individuals actually only creates a priming effect and doesn't complete the response. And so overall, we see a lower amplitude in terms of an antibody measure, like an antibody concentration. And then the fear is over time is that declines more severe than the general population, necessitating a third dose. And we're always trying to get these amplitudes as high as possible to reference groups as best as we can expect. And so to do this study, essentially we required massive partnership. And so we partnered with Schlegel villages St. Joseph's Elder Care, the Health Sciences North Research Institute, and recently Shalom Village and in Amica to look at retirement homes. And essentially we targeted and recruited a huge number of long-term care and some retirement homes now, and indeed probably some assisted living communities in mostly Southern Ontario to do this study. And so we really thank our partners for being involved in this. They are equal partners in this work. We could not have done it without them. Our research coordinators, and of course the residents and families that consented to do this at a time when it still remains very, very difficult and very trying in long-term care. When we say an antibody-mediated immune response, we're kind of measuring these three features. Number one, how many antibodies are made to the vaccine, and so we can count these. They don't give us a full picture, but they tell us. Obviously, we're making antibodies to the spike protein with the vaccine. And then we want to know, do these antibodies actually recognize the receptor binding domain of the virus? Because sometimes you can create a lot of antibodies, but they may not be the correct antibodies, which is antibodies to the receptor binding domain, which is sort of seen in this cartoon here as a feature of the spike protein. And then the third and most important is, how effective are they? And so this is defined really as neutralization, but Don describes this very sort of friendly as sticky. So how sticky are the antibodies? Can they actually stop the virus? And so this is maybe a clearer picture, so still very cartoonish, where you have the virus and you have the spike protein of the virus, which is what attaches to our host cells through the receptor binding domain and essentially attaches to the ACE2 receptor. And what an antibody is supposed to do is basically, you see it here by this sort of Y-shaped little schmutz, is stick to the receptor binding domain, therefore not allowing it to attach to host cells, rendering it essentially not inert, but not harmful. To the first picture, and the pictures will look a lot like this. We want to understand how many residents responded to the vaccine by understanding how many antibodies were made to the vaccine. And this shows every single dot is a long-term care resident. The squares are a reference population just for interest of your eye. And we classify the distribution of the response by green, yellow, and red. Green is those that make as many antibodies as we have seen in the comparison cohort, which is healthcare workers in the Hamilton region, which range from ages of about 21 years old to as old as 68, of varying health. So not necessarily a healthy cohort, but a cohort we expect a good response. And this green zone here essentially defines the confidence interval of their distribution. The red zone is essentially those where we can't detect any antibodies, which we see in some residents. And this, I should clarify, is at about 4 months post-second dose of the vaccine. And the yellow zone is those in between, where we probably don't have a lot of concern for residents who responded in the upper yellow zone, but have a lot of concern to those in the lower yellow zone. And so this is the picture. It gets more clear as we move on. Now, how many residents made the right antibodies? And you see already the distribution changes at that second most important question. Again, in the green zone, we see about half of residents, and then in the yellow and the red zone, we see approximately the other half at about 4 months post-second vaccine, which is concerning. And more individuals are falling into a zone of concern, and most concerning, a zone of red, which essentially means that there were no detectable correct antibodies, so antibodies made to the receptor binding domain. That's concerning because you can't have an immune response if you don't have the correct antibodies in any kind of concentration. And some of this jittering is really just for the purpose of the figure. The third question, how sticky are the antibodies? And so can they stop the virus? At McMaster University, we're very fortunate to have colleagues and facilities that are able to do this very specific assay, which is a microneutralization assay. And what it essentially is a level 3 lab where they have the full getup like you see in movies, where they have live virus exposed to individual resident serum in an assay, and they essentially have them battle it out and measure its neutralization capacity, which is really classified as the number of dilutions you can make to the individual serum where they still essentially neutralize 50% of the virus. And so that's why higher is better in this case. We define a red zone here, which is essentially the standard established and often used from and published in Nature Medicine that was based off of the trials, the vaccination trials, some very large convalescent cohorts where they understood this measure and then they tracked going forward to look for clinical outcomes like infection, severe infection, and death. And what they eventually found was at this level, which is about 20% of the convalescent neutralization level, was highly associated with infections as well as serious infections and death. And so what we overall find is that overall for the original variant, which is the Wuhan variant, you see here on the left, and in the lab we also have the Beta or the South African variant. What we note is that after 3 months, about 20% overall of residents fall in this area of concern where they're not predicted to be protected to the virus, severe infection, as well as death. And so the good news is, of course, is that for the majority, 80% at that level, at that point, 3 months post, they were performing quite well in terms of their neutralization. The bad news was that for some, it wasn't where we would hope it would be. How long do they last? And so this is a bit breaking. And based off of this figure, essentially, and corroborating evidence from teams that are doing some of the very similar work across the country, there was a recommendation for the third dose. A lot of evidence that an expectation that a third dose would be required in some populations, and this was enough to recommend it immediately now, and this is reporting on about 3 or 4 months post second dose, and at the time that the decision was made, we are now at about 5 to 7 months post second dose. We had a measurement in residents that occurred at about 5 to 6 months post second dose, where we measure on the very same scale seen on the right around their antibody neutralization. And what we found is around 2 and 4 months, we have a picture of about 20% falling in the red category, and between 4 and 6 months post, which is now closer to when the decision was made, although the decision was mostly made off of these data, we have 70% of residents falling into this red zone, which is the prediction that they are at risk of the infection. Some of this was pretty apparent over the summer, at least we thought so, where the clinical evidence in the field was that over the summer we were already seeing breakthrough infections. We were already seeing some fairly concerning, I'd say always very concerning outbreaks. And we did not expect this, or we shouldn't expect this, given that over the summertime we had very, very low circulating virus. And so that we saw it sort of confirmed the picture at 2 and 4 months, but that we're seeing a larger number falling into this red category at about the time that the dose was approved, I think suggests we made the correct decision. That is in comparison to colleagues who are doing similar studies in terms of measurement in healthcare workers, where you see a very, very different distribution where many of them are falling into good categories at around 6 months. And this looking at an earlier on cohort of individuals who recovered from their infection, who we call convalescent cohort, which was essentially an incomplete response. We see that here as well, that they look a lot like our cohort, which is at 4 and 6 months, some of them having an incomplete immune response, or certainly not as good as we'd hope. We've also looked at the vaccine type, and they matter. And so Moderna versus Pfizer, what we find is that at about 5 months, 20% of Moderna recipients fall in the red zone compared to about 60% of Pfizer recipients. This has been shown across various studies. And so the quality quantity is higher as well as longer for Moderna. This is not totally unexpected because Moderna has 3 times the mRNA or the antigen that creates the response, which is a lot like influenza vaccines that are available to long-term care residents that have a lot more antigen, which essentially is the stuff that makes your body react to it. And as well as a longer interval between doses, which is generally good, was a bit longer for the Moderna than the Pfizer, according to manufacturer recommendations. And so right now the question is what is the optimal vaccine use? And we are starting to be able to look at this where we have the natural experiment of different types of vaccines being used. Do extra doses help? We think so. Certainly we see this in our other cohorts where we see a very predictable response, sort of like that line graph I showed you in healthcare worker cohorts, where we have some evidence sort of in the study. This looks at creating the correct antibodies, where we have residents that had a previous probable or confirmed infection. So essentially 3 doses, 1 natural, we see a much better response. Some of that is biased by the fact that, of course, they survived that infection, which probably happened earlier on in the pandemic. So there's a bit of a healthy bias here as well. Ongoing research, we're monitoring this as well as other studies are monitoring this, monitoring breakthrough cases for those that are fully vaccinated, those that are partially vaccinated, which you sort of see here. Essentially we have some of these coming in now where we're able to look at them. We're looking at cellular immunity as well. The antibody-mediated immunity is really a piece of it. The cellular immunity is the other bit, and very interesting, and we're exploring that. The work now is also to understand correlates of protection, which is really what do we know about residents who do and do not respond to the second dose of the vaccine as well as the third dose? Are there drugs, medications, or health conditions that can help us sort of understand why and also prioritize these residents going forward if some still don't have the effective response that we're hoping for. As I finish, I want to acknowledge just this absolutely massive team that we have working on this, some of which are shown here, many which are not, who are working very diligently in very difficult circumstances from the beginning to really make this study happen. And so we really appreciate them. And thank you very much to the RIA for inviting us. Visit our website if you want to hear more, and I'm happy to take questions.
All right. Thank you very much, Andrew. It's always so exciting to hear about this research sort of results as we're getting them and following the progress of this study for sure. There are a couple of questions that have come in. You did talk a little bit about the time between the vaccinations and so on. The question is, do you know what the ideal time is between vaccines for older adults?
It's a great question. It's very difficult for us to know the ideal time, or certainly I don't have the correct answer there. I haven't seen data that shows that. Essentially what we have is the manufacturer's guidance around, because obviously, recall that we really needed to get the vaccines out as fast as possible. So the scientific question was, how close can we put them together without really hurting the immune response? And that came out of the trials. And that was essentially the manufacturer's recommendation. In general, waiting longer is definitely better because you kind of let that prime soak in a bit longer. In general, that's the case. Here, I don't think we know the ideal. And in this study, we won't be able to say precisely what the ideal is because the dosages and when they came were to manufacturer schedules, so we don't have a lot of variation.
Right. Okay, that's great. An interpretation question. There was a graph that you showed, sort of the green, or red, yellow, green graphs. What was along the x-axis on those ones? Specifically, the question is the one about how many residents responded to the vaccine. What would be along the x-axis there?
The x-axis was either the vaccine type, where we compared Moderna in the last figure and Pfizer, or it was the particular cohort. We're comparing our cohort to healthcare workers, or a convalescent cohort of those that recovered from COVID earlier on in Hamilton from other studies, which really gives us a sense, it's sort of a North Star in terms of comparison. And those groups essentially define what the green category is, according to sort of international standards of understanding neutralization. Beyond that, some of the figures you see that there's a bit of jittering of the circles. That's essentially just so you can be able to see the variation, like see the circles away from each other, because otherwise they'd be stacked on top. They would look like one messy line.
Right. So the ones where it looks sort of like a funnel, that's just so that we're able to see the number.
Correct.
Of
the more you have in each category, the more it sort of funnels it out. So it's not suggesting anything more better, just that you can see the density of residents that are represented by those circles.
Okay, great. I'll just continue along with the questions here. Were there any participants who had virtually no immune response to the third dose?
You know, we don't know that yet. And so the third dose has essentially recently been rolled out to almost all, if not pretty much all, long-term care homes. In fact, I think it's totally complete now. I don't think there's a one in the province now that hasn't complete, certainly not in our group. Mm-hmm. And right, we have to wait between 3 and 5 weeks, well, about 3 weeks or 4 weeks as soon as we can to get that essentially what is a dried blood spot from residents who consent that we do that. And only then, after we process that information, will we know what the outcome has been. I think we can expect it. What we're hoping for is that those that are in the red go into the yellow, high yellow, maybe green. Those in the yellow go to the green. Those that were in the green stay in the green.
One of the exciting things about being sort of getting the results as they're coming in, right? We're always learning more and more. When you talked about the increased breakthrough infections, were they symptomatic? And was there increasing poor outcomes, such as hospitalization and so on, associated with those breakthrough infections?
I haven't analyzed the data or seen analyses of that in particular, so I'm relying on essentially anecdote. What I would expect to see is that the case fatality rate for those breakthrough infections would be much, much less. Indeed, the outbreaks that have occurred on the outbreak level, there's been much less sort of severe outcomes associated with it, symptoms being less severe or not apparent, where we would typically see a proportion of deaths occurring, we're seeing far, far less now. So that's really great. And that's really the value of having the vaccine in place, is that there is some immunity there, and that really accounts for why we don't see the same kinds of terrible outcomes that we did early on in the pandemic.
Okay, great. I think we have time for one more question before we're at time. The question is, I'm intrigued about the upcoming work to look at correlates of good versus poor immune response. What are some of the suspected correlates?
You know, I don't know that we have suspected correlates. We've looked at this a bit anecdotally by looking at charts, et cetera, and doing chart reviews for those who didn't have the response that we would like, to try to identify some hypotheses. And there seems to be maybe some hint of chronic kidney issues or kidney issues. There seems to be some hint of potentially aspects of frailty and immune frailty, and there's different ways to measure that. And what we didn't get a strong signal for was sort of immune modulating medications associated with arthritis, et cetera, things that lower your sort of immune function. We haven't detected that yet, but we haven't been in a position to really do a serious analysis on it yet, because we are still bringing in the full picture of factors to be able to do that.
Sure.
So that remains very preliminary.
All right, great. Well, there are still some additional questions, but I know that we're at 2 o'clock, and so like I said at the beginning, we will send out some responses to all of the questions that have come in. That'll be posted on our website shortly in the next week or so. But in closing, just thank you so much, Andrew, for sharing these exciting results and for sharing some preliminary thoughts about what next steps might be and what this might mean in the future as well. We really appreciate your time and speaking with us this afternoon.
Well, thank you, Susan. It was a pleasure.
That's great. So for everybody on the call, thanks so much for joining us. We will be having another webinar on November 16th. Dr. Al Power from Rochester will be talking to us about how to create kind of a sense of belonging and trust and comfort in long-term care and retirement living. So registration for that one will open on October 27th. But with that, we'll close for today and hope to see you all again soon.